One of the most powerful techniques in bacterial genetics is the ability to identify genes by transforming bacteria with bacterial genomic libraries. All you need is a strain with a mutant phenotype due to a mutation in a single gene. Suppose you have an E. coli strain that is auxotrophic for arginine because it has a loss-of-function mutation in a gene required for arginine biosynthesis. You can clone that gene by transforming a genomic library made from wild-type E. coli into the arginine auxotrophs. Here, the genomic library is made in plasmid vectors with an ampicillin resistance gene as the selectable marker. The auxotrophs are growing in minimal medium supplemented with arginine. After mixing the library plasmids with the mutant bacteria, you can select for a transformance by plating the cells on medium containing ampicillin. If you omit the arginine supplement from the medium, the only colonies that will appear are bacteria transformed with a plasmid that enables the cells to make arginine. That plasmid most likely contains a wild-type copy of the E. coli gene that is mutant in the auxotrophs. The first step in checking that you've really identified the gene that is mutant in the auxotrophs is to compare the base pair sequence of the wild-type gene in the plasmid to the copy of that same gene in the auxotrophs. If the gene copy in the auxotrophs has a mutation that is likely to block its function, then you've likely identified the right gene. You can do this easily. First, you sequence the gene in the plasmid that rescues the auxotroph's mutant phenotype. Next, you use this sequence to design PCR primers that will amplify the corresponding gene from the genomic DNA of the auxotrophs. Finally, after you perform PCR, you sequence the amplification product and compare it to the base pair sequence of the wild-type gene in the plasmid. A nonsense mutation, for example, is likely to block gene function. Gene targeting is another experiment you can do to make sure that you've cloned the correct gene. In gene targeting, you use the gene in the plasmid to make a mutation in the corresponding gene in wild-type E. coli. First, you use cloning technology to make a construct where an ampicillin resistance gene is flanked by the 5' end and the 3' end of the gene in the plasmid. Linear DNA molecules transformed into bacteria will recombine into the wild-type copy of the gene, making a loss-of-function mutation. If the gene in the plasmid corresponds to the mutant gene in the arginine auxotrophs, the new mutant strain you created by gene targeting should also be an arginine auxotroph. This scheme for cloning a gene corresponding to a mutant phenotype will work if your starting strain has a loss-of-function mutation in that gene. The wild-type gene on the plasmid provides the function that the genomic DNA in the mutant bacteria lack. But suppose the mutant allele causing arginine auxotrophy has not lost its function but has gained a new function. For example, suppose the mutation is in a gene that normally makes a protein with nothing at all to do with arginine biosynthesis. The mutation causes the gene to make a mutant protein that degrades an arginine biosynthesis enzyme and thus blocks the arginine biosynthesis pathway. Transforming the auxotrophs with a wild-type copy of that gene will not enable them to make arginine, so we cannot identify gene X this way. What can we do? To identify a gene corresponding to a gain-of-function mutant phenotype, you need to do everything we did in our previous scheme, but in a sense backwards. Instead of identifying a plasmid that restores the wild-type phenotype, the ability to make arginine, to the mutant strain, you seek to identify a plasmid that imparts the mutant phenotype, the inability to make arginine, to a wild-type bacterial cell, and that plasmid should contain the mutant gene X. Begin by making a genomic DNA library not from wild-type E. coli, but from the strain with the gain-of-function mutation. This way, one of the plasmids in the genomic library will contain the mutant allele of the gene you want to identify. Next, you transform the mutant library into wild-type bacteria. Transformants are selected by plating on medium containing ampicillin, and then screened to identify arginine auxotrophs. An arginine auxotroph must contain a plasmid with the gain-of-function mutant allele. To check that you've cloned the right gene, you can amplify the wild-type copy of the gene in the plasmid from normal E. coli and compare its base pair sequence with that of the gene on the plasmid. If you've identified the correct gene, then the gene in the plasmid ought to contain a mutation likely to lead to gain of function, such as a missense mutation.